Good afternoon, all. So, our next topic from the methodology and instrumentation is about biological safety cabinets. So, this is the picture of the three types of biological safety cabinets which are used in our laboratory. Introduction. A biological safety cabinet, short form BSC. It is also called as biological safety cabinet or microbiological safety cabinet. It is an enclosed ventilated laboratory workspace for safely working with materials which are contaminated with pathogens requiring defined biosafety levels. So we are usually working in this workspace with contaminating pathogens or pathogens itself. Several different types of BSC exist, which are differentiated by the degree of biocontainment required. And this biological safety cabinets first became commercially available in 1950. Now, next is the purpose. The primary purpose of this biological safety cabinet, it is to serve as a means to protect the laboratory worker and the surrounding environment from pathogens. All exhaust air is HEPA filtered because it possesses a HEPA filter inside the cabinet. And it exists in the biological safety cabinets, biosafety cabinet, removing harmful bacteria as well as viruses. This in contrast to a laminar flow clean bunch, which blows unfiltered exhaust air towards the user, and it is not safe for working with pathological agents. So in the laminar airflow, whereas with a clean bench, there is a chance of blowing unfiltered exhaust air. So we will get exposed to the pathogenic organs. So to protect us from the pathological agents, we are using biological safety cabinets. Neither are most BSCs safe for use as fume woods. So it is safer than fume woods. Likewise, the fume wood fails to provide environmental protection that HEPA filtration in the biological safety cabinet would provide. However, most classes of BSCs have a secondary purpose to maintain the sterility of materials inside the cabinet also. Biohazards and biosafety levels. The word biohazard is a contraction of the words, that is biological and hazard, and it is defined as an infectious agent or part thereof presenting a real or potential risk to the well-being of man, animals or plants directly through the infection or indirectly through the disruption of the environment. Biosafety levels 1 to 4 were established by the CDC and the National Institute of Health and are the combinations of laboratory practices and techniques, safety equipments and facilities. All of these levels are appropriate for the biohazard posed by the agents used and the laboratory activity. So there are 4 types, but here I am mentioning about only the 3 types of biosafety cabinets. Next is the classes. The US of Centers of Disease Control and Prevention classifies the BSCs into 3 classes. 3 classes, and the types of BSCs within them are distinguished in 2 ways. The level of personal and environmental protection provided, the level of the product protection provided. So, actually this class 1 safety cabinet, it is the most basic one which provide personal and environmental protection only. When properly used, biological safety cabinets have been shown to be highly effective in reducing laboratory acquired infections and the cross contaminations of the cultures. So 3 classes are present. First, we can discuss Class 1 Biological Safety Cabinet. The Class 1 cabinets provide personal and environmental protection, but no product protection. In fact, the inward flow of the air can contribute to the contamination of samples. Inward airflow, which can be maintained at a velocity of 75 per minute or 0.3 meter per second. These biological safety cabinets are commonly used to enclose specific equipments such as centrifuges or the procedures. For example, aerating cultures that potentially generate aerosols. The BSCs of this class are either ducted, which have been connected to building exhaust system, or unducted, recirculating filtered exhaust back into the laboratory. So it can be either ducted or unducted. Laboratory personnel have specific training in the procedures conducted in the laboratory and are supervised by a scientist with general training in microbiology or a related science. Next is the working mechanism. So this is the side view of the Class 1 safety cabinet. This shows potentially contaminated air, and this one is the HEPA filter. This is the room air. Room air is drawn in through the front opening at a minimum velocity of 0.38 meter per second. The front opening also allows the operator's arm to reach the work surface inside the cabinet, while he or she observes the work surface through a glass window. The windows can also be fully raised to provide access to the work surface for cleaning or other purpose. The directional flow of air whisks aerosol particles that may be generated on the work surface away from the laboratory worker, and they are discharged from the biosafety cabinet through a HEPA filter. So it possesses a HEPA filter also, which removes the contaminated air. Second one is about Class 2 Biological Safety Cabinet. This is the Class 2 Biological Safety Cabinet picture. And this Class 2 cabinets provide both kind of protection of the sample and the environment, since makeup air is also HEPA filtered. Mainly there are 5 types. Type A1, formerly called A. Type A2, formerly it is called A by B3. Type B1, Type B2 and Type C1. Each type's requirement are defined by NSF International Standards 49, which in 2002 reclassified AB3 cabinets into Type A2 and added the Type C1 in the 2016 standard. About 90% of all biosafety cabinets installed are Type A2 cabinets. Next is the principle of operation used is, it is a motor driven blowers or fans mounted on the cabinet, which helps to draw the directional mass airflow around the user and into the grill, protecting the operator. The air is then drawn underneath the work surface and back to the top of the cabinet, and this can pass through the HEPA filter. A column of HEPA filtered sterile air, it is blown downward over the products and helps to processes to prevent what? Contamination. Air is also exhausted through HEPA filter, and depending upon the type of Class 3, the air is either recirculated back into the laboratory or pulled by an exhaust fan. Next is Next is about different classes of the class 2, not different subtypes of class 2 biosafety cabinet. Type A1. Type A1 cabinet, formerly known as Type A, has a minimum inflow velocity of 75 feet per minute. The downflow air considered contaminated spills just above the work surface and mixes with the inflow. This air is drawn through the ductwork up back of the cabinet, where it is blown into a positive pressure contaminated plenum. Here, the air is either recirculated through the HEPA filter or exhausted out the cabinet. Sizing of HEPA filter and the internal damper are used to balance these air volumes. And this type is not safe for work with hazardous chemicals even when exhausted with thimble or canopy to avoid the disturbing internal airflow. Next is about Type A2. The Type A2 cabinet, formerly designated as A by B3, it has a minimum inflow velocity of 100 feet per minute. A negative air pressure plenum surrounds all the contaminated positive pressure plenums. In other respects, the specifications are identical to those of a Type 1 cabinet. The Type B1 and B2 cabinets, they have a minimum inflow velocity of 100 feet per minute, and these cabinets must be hard ducted to an exhaust system rather than exhausted through a simple thimble connection. Their exhaust system must also be dedicated, in contrast to the Type A1 and A2 cabinets. The Type B biological safety cabinets use single pass airflow in order to avoid or control hazardous chemical vapors. Type B1 cabinets spill the airflow so that air behind the smoke split is directed to the exhaust system, while air between the operator and smoke spill mixes with inflow air and it is recirculated as downflow. Since exhaust air is drawn from the rear grill, the CDC advises that work with hazardous chemicals can be conducted in the rear of the cabinet. This is a complicated since the smoke split is an invisible line that extends the width of the cabinet and drifts as an internal HEPA filter load with particulate. Next is about Type B2. Type B2 cabinet is expensive to operate because no air is recirculated, and this type is mainly found in such applications such as toxicology laboratories, where the ability to safely use hazardous chemicals is important. Additionally, there is a risk that contaminated air would flow into the laboratory if the exhaust system for the Type B1 or B2 cabinet were to fail. To mitigate this risk, cabinets of this type generally monitor the exhaust flow, shutting off the supply blower and sounding an alarm if the exhaust flow is insufficient. The Type C1 BSC was born out of necessity to control infectious materials, chemical hazards, reducing operation cost and add flexibility in modern laboratories. This Type C1 moves air by mixing inflow air with the air in the column of downflow air. So, this C1 differs from a Type A that it can use this single pass airflow, and when installed in ducted operating mode, it can protect from the hazardous chemistry like Type B's. The C1 also differ from Type B in several ways. What are the difference? It does not require a hard connected exhaust system and a blower to operate, and also it can run for an extended duration to increase the operator protection during the remote exhaust system failure. The type C1 BSc biosafety cabinets can run without being connected